All right, it looked as if the recording didn't work properly in the lecture, so I'm going to give an abbreviated version of the lecture here. Actually, it may not be that abbreviated. We'll see how I do for this first lecture for Introductory Biology for Health Sciences. If you have any questions, I understand that this is a technically difficult time for all of us, so please send me an email. My contact information can be found on this next slide here. So my name is Dr. Evan Christensen. I'll be your instructor for the semester for BI105. My email is here on the screen and my office is in Psi 301B. I'll be holding office hours throughout the semester on Mondays and Tuesdays from 10:30 to 11:30 a.m. and then also on Wednesdays from 3 to 4:30 p.m. These are open office hours. That means you can come to them for any reason, up to and including you just want to talk to me. You're welcome to come for that reason as well. But if you have questions, general questions about the class, things like that, feel free to come to an open office hour, these listed here. If you want to make an appointment, these appointments should be made for sensitive matters. If you want to talk about, for example, your performance on an exam, or if you want to talk about your personal life or something that's affecting your performance, please book an appointment with me rather than coming to open office hours. That way we can have a more personal conversation. Open office hours and appointments will be held over Zoom, and to book an appointment in the appointment booking software, you should go to this URL, echristia.youcanbookme, and that will give you available appointment times. Okay, for this course, we will be holding the exams and the quizzes using ExamSoft. ExamSoft is an online testing platform. You can sign up by going to examsoft.com. You will be given exams through your computer, so you can use your laptop, either Mac or Windows PC laptops, or you can use an iPad. Chromebooks cannot be used for ExamSoft. We can discuss accommodations for that as we go. We will also be using an online learning platform called TopHat. TopHat will be used for in-class participation, in and out of class homework, textbook, to deliver the textbook, and for attendance. TopHat does have a cost. It is $40 for the textbook, as well as $26 for TopHat for one semester, or there are subscriptions that can give you a better cost. That being said, I think these numbers might actually be lower. I think it might be $20 for TopHat for the semester. The rep is working out something on that. You can use one active TopHat account for all of the courses that require TopHat. So if you take another course with me, for example, in the spring, where we will use TopHat again, you can use the same account. Unexcused absences in this course are going to be according to a late work policy. In-class assignments will have a 24-hour window that includes exams. Reading assignments, homework assignments, etc. will have a 24-hour or longer window to complete them. Because this class is being given as a hybrid class, there's basically no reason why you should ever turn in late work. You will have lots of time to complete it, and it's not bound by the time that you actually would attend class directly. So there should be no makeup assignments in this class. If you have an emergency or something comes up that qualifies under emergencies, let me know and we can work out a solution, but makeups are broadly not offered in the class. Makeup labs follow the same guideline. There will be no makeup labs except for in exceptional circumstances, and makeup labs can only be done online. There will be no in-person makeup labs this semester. Note that if you have more than 2 missed labs, that is an automatic failure of BI105. So make sure that you attend lab when you're supposed to. Please talk to us if it begins to become an issue. Note that there are certain ways that you can have excused absences. The first is a religious observance. The second would be an illness or documented emergency, something that you can provide documentation for. And then the third would be sanctioned school functions like sports or potentially interviews. Please contact us if you have problems with or if you have an excused absence. Note that excused absences should be able to be well in advance unless they are an emergency, in which case you should be able to provide documentation. So just let us know, for example, if you have a religious holiday that comes up some point during the class, just let me know in advance so that I can accommodate you. Academic conduct, I'm not going to go over in detail. The academic conduct code is available at this link. Please don't cheat in my class. It'll be a problem if you do, and so it's better just not to. The course grading is as follows. In the lecture class, you will have 4 exams, 3 sort of midterm exams or during class exams, and then 1 final worth 20%. And then in-class assignments and homework are worth 15% of your total grade. In lab, there is no exam in your lab class. The entire grade is made up of pre and post-lab assignments, as well as in-lab assignments, and then 1 final project. Usually I get a question at this point whether or not the final exam is cumulative. The answer is that it is sort of functionally cumulative because the material builds on itself as we go, but it is not sort of formally cumulative. I won't ask you questions that are directly about material from exams 1 through 3 on the final exam. However, I can require that you know that material to be able to answer a question. So I might include question material from exams 1 through 3 in the question on the final exam, so that it would be very hard to answer it if you didn't understand it. Note here also that a failing grade in the lab portion of the class will fail the lecture portion of the class as well. So if you do not earn a 60% or higher in lab, you will also not pass lecture. Because of the exam structure, our class is split into several modules. Module 1 is about chemistry, macromolecules, and cells. Modules 2 through 4 will cover the remainder of the course, but this is the sort of schedule for the next couple of weeks here in this class. We're going to talk about the scientific method, chemistry for biologists, biomolecules, cells, and membranes. Okay, so usually here I would pause for any questions. In the class, the only question that came up was that one about the final exam and whether or not it was cumulative. So we'll launch right into the course introduction here. The first question to ask in a course like this is, why are you taking this class? Or why are you in this class is a sort of spin way to phrase this question. You might say because I'm studying for medicine, but really the answer is because we have made so many advances in medical biology, in biological sciences, that people don't die young much anymore. It's really quite uncommon for people to die young. Whereas if you go even just 100 years back in time, many people, there were 300 cases per 100,000 population of measles in the United States in 1912. The same goes for polio. As recently as 1955, there were just about 40 cases per 100,000 population. And polio, in most cases, was a terminal or very seriously debilitating disease. So the reason why you're in this class is because you didn't die of a disease when you were quite young, and that's a good thing. We're going to explore the biology behind some of these things and how we've gotten to the point that we have biologically now in this class. Shortly though, biology is the scientific study of life. What that means is we're studying living things at all of the different ways that you can study them. Here we're going to be talking in this introductory PowerPoint about how biology can be performed at different levels of organization, how biology can be concerned with multiple timescales, how it can be studied as either a basic or as an applied science, and then also how it's unified by several fundamental principles. Starting with the levels of organization. What I mean by levels of organization is that biology can be studied from the smallest component of biology, the atom, the individual atom, all the way up to the largest component of biology, the entire biosphere that we know of. So starting with atoms, moving up to molecules, which is the interaction between atoms, moving up to cells, which is the interaction between molecules, and tissues, which is the interaction between cells, et cetera, et cetera. We can move through the levels at which you can study biology, and all of these are equally a biological study. So somebody might study the macromolecules or molecules of biology. This would be like a biochemist. Or they could study all the way at the top, they could study the entire biosphere. This would be an ecologist, someone who studies a very large scale of biology. But all of these are biological study. So biology is the study of life that can be conducted at many different organizations, but it can also be conducted at different time scales, at different amounts of time. So here we can see changes that occur quickly. So going from a resting state to a fighting state for a lion, that's a very quick, acute change. We might also think of things that occur chronically, like getting a sunburn or tanning. This takes a long time. It doesn't just happen right away. Or in the case of Chris Pratt here, getting very fit for his movie roles. This is something that also doesn't occur very quickly, but it takes a long time to happen. Then we have periodic changes, things that occur at regular intervals, like you get tired every day because the sun goes down. So your exposure to the sun changes your energy level. Or this rabbit on the bottom right changes its coat according to the seasons. And this is a periodic change, something that occurs at a regular interval. We also have developmental changes, which occur throughout an organism's lifespan. Here we have a monarch butterfly going from a caterpillar to a butterfly. And this monarch is going through these developmental changes, one stage at a time, according to a pre-programmed set of circumstances. So the caterpillar already has built in within it the developmental stages that will bring it to the sort of full butterfly form. Also, developmental changes are irreversible. They don't go backwards. And that's how they are different from chronic changes. One, they are pre-programmed. They're going to occur even if you don't get exposed to environmental conditions, usually. And also, they don't go backwards, unlike, for example, a tan, which can fade. Finally, we have evolutionary changes. These are changes that occur to a population of individuals and that occur at the level of their DNA. So these are permanent changes that can be inherited by future generations. And all of these are timescales at which biology can be studied. Next up, we have biology being able to be performed as a basic or as an applied science. What this means is you can study biology just sort of for its own sake. So biology then can be studied like just studying animals directly, for example, or studying chemical processes directly. There's no goal in terms of how this impacts humans. It's just an interest in the science. These are driven sort of by a general desire to just know more about the thing you're studying. You just want to know more about it. That's all basic biology is. Applied biology is when you take that knowledge from basic biology and you're driving your investigation to specifically improve the life of humans. So you could think of this as something like health science research, right? Studying how a drug affects human physiology. That's very directly related to human flourishing, and so that would be an applied biological study. Usually, applied biology depends on the discoveries that are found in basic biology. So when you're studying basic biology, you are sort of giving tools to the applied biologists later. Most people understand this maybe a little bit better when we talk about physics and engineering, right? So physics is how we study the natural world and its physical relations. And then engineering is how we make use of that. And so engineering here is the applied science and physics is the basic science. In biology, usually this is health science is the applied science, and sort of basic biology like ecology is the sort of more basic version. One example of this is the way that we studied these things called ACE inhibitors. An ACE inhibitor is a blood pressure medication, and a blood pressure medication that lowers blood pressure. Well, this was discovered in the Brazilian arrowhead viper. So the Brazilian arrowhead viper is a snake that lives in the Brazilian Amazon rainforest and has this ACE inhibitor chemical in its venom. But we weren't looking for a blood pressure medication when this venom compound was discovered. This was just something that the scientists who were studying the pit viper were interested in. And then when they discovered that it lowered blood pressure, different scientists then tested this as a medication, and now it's a very common medication used for blood pressure lowering. Okay, so we have biology performed at different levels of organization, at different timescales, and as a basic and applied science. And this might make it seem like every biologist is operating sort of differently, like everybody is using different things to study biology. But actually, biology is unified by several fundamental principles. So we're going to go through these one at a time. Biology is a sort of emergent property, right? So you may have seen these images online or something like that, where it's a timeline and it shows you how the sciences are related. So mathematics is the sort of most basic way that we do science, right? And then you say that physics is applied mathematics, and chemistry is applied physics, and then biology is applied chemistry. And what that might make it seem like is the case, it might make it seem like biology is like simpler or sort of downstream of physics. But actually what this means is that biology has lots and lots of properties that emerge from the complex interactions of chemicals. So actually biology is sort of more complex and less predictable than chemistry is. So let's look at this one at a time. So if you start with a human person is a very complicated individual organism, right? You have a hard time predicting what my next behavior will be because you're not me and you can't predict all of the chemical interactions that are occurring inside of me at any given time. Right, all of those chemical interactions are happening inside of the brain, which are actually happening inside of neurons, which are actually chemicals. As you can see here on the bottom left, chemical interactions that are occurring all the time. And each of these chemical interactions has a defined outcome. But when you combine them all together, this forms like this really complex interaction that's hard to predict. And as we go up in the scale, we see that this molecular machinery generates additional properties as we go up. Okay, so another one of the properties of biology. So the first one was that emergent properties come from systems. The next one is that living organisms interact with their environment. So here you see plants that are orienting themselves towards the sun. They're reacting to the sunlight coming through the window. You might not think of plants as interacting with their environment, but they do all the time. They take in sunlight, they use that sunlight to generate energy, they face their leaves or their heads towards the sun, they take in water from the ground, etc., etc. Plants, just as well as animals, interact with their environment. And in fact, all living things do. Living organisms also use energy. This is part of their interaction with their environment. They use energy to interact with their environment. So plants use energy in the form of light to photosynthesize, to produce sugars, which they then break down to create ATP, which we'll learn about later. And they use this ATP to orient themselves or to create new proteins, etc. Animals eat plants or they eat other animals, and this is also taking in energy. So living organisms use energy in this way. The structure of living things is related to their function. So if we take a look at this anteater, there are all kinds of things about this anteater that are directly related to its function. The first is it has quite large claws, and these claws are used for digging at anthills. It also has an elongated snout, and you can't see it, but a very elongated tongue. And these two things are used to get at the ants that it needs to eat. So the structure of its claws and of its tongue and of its nose are all sort of related to the function that it does. Okay, cells are the simplest units of life. What this means is that the cell is the smallest self-contained unit that is living. Viruses are smaller than cells, but we don't say that viruses are alive. So that means that cells are the simplest unit of life. You are made of cells. You are, or I should say, you are composed of cells, and you are also composed largely of cells that are not your own. So by number, your body is about 10 to 1 composed of non-human cells. Now, by weight, you are more human than you are bacterial, for example, but by number, there are actually more bacteria in your body than there are human cells. Organisms grow and develop. We sort of already mentioned this, right, when we talked about the organisms being able to be studied in their developmental timeline. Well, organisms grow and develop from young to old. All organisms do this. They at least grow, but most of them also change over the course of that growth, meaning development. You can see here the difference between a baby penguin and an adult penguin, right? They look different. They have different feather colors. They're different in size. One of them is sexually mature and the other is not. All of these things are developmental processes that occur, and all organisms either grow and or develop. Also, all organisms are united by their common genetic heritage. So all organisms use DNA as their genetic information to store the information that allows them to make themselves. A question that I asked in class here is, why do offsprings resemble but not identically resemble their parents? And the answers we gave were, number one, parents share the genetic responsibility for their offspring. Both the mother and the father contribute to the genetic heritage of the offspring. So an offspring is sort of half and half, mother and father. But also there are other things that could be introduced. The interactions with their environment. For example, a child might be thinner or fatter than their parents because of their diet. They might be more or less fit. They might have scars that a parent doesn't have. They might lose a limb, for example. There are all kinds of ways that a child could be different from their parents. In addition, there could be mutations in the DNA that lead to a difference in the phenotype and the appearance of the child. Or there could be recombination that occurs when the parents pass on their DNA, which result in a slightly different looking child. Okay, two more. Almost finally, all species are related by their evolutionary history. So all species, because they share the same DNA, are related to one another. So DNA is used by all organisms, from bacteria to giraffes, as you see on the picture here. And these organisms all are related. We're not going to talk a lot about evolutionary history or the classification of animals, etc. You're welcome to take Bio 309, that's evolutionary biology, if you're more interested in this topic. But we will talk a little bit about how evolution affects health science as we get towards the end of the class. Okay, last but not least, biology affects our society, and our understanding of biology affects our society. On the left-hand side here, you see corn. Corn is a genetically modified organism. Even non-GMO corn is a genetically modified organism in the sense that we selected over thousands of years, or I should say the Native Americans selected maize, the plant that gives rise to corn, selected it to have larger ears and give more fruit. And this is a genetic modification. Also, as you can see on the right-hand side, disease affects our society quite severely. It's the reason I'm giving you this lecture this way right now, is because a disease has drastically affected our society. So biology in the form of genetically modified organisms, in the form of health science, in the form of all kinds of different topics, affects the way that we go through our society. Okay, in this class, when we talk about biology, we're talking about hypothesis-driven scientific inquiry. What I mean by that is we're talking about a specific type of biology. We're talking about biology that asks questions and answers them with experiments. So that's what we're going to be talking about in the class. Hypothesis-driven science starts with observations and then leads to questions. So for example, if you are looking at a bunch of animals drinking from a water hole, you might ask the question, do all animals need water to drink? This would lead you to formulate a hypothesis based on your current understanding. So you might see all of the animals that you've ever seen drinking water, and that might lead you to say, yes, all animals need water to drink. This positive statement is your hypothesis. A hypothesis isn't a question, it's a statement. You're saying, yes, all animals need water to drink. And then you make predictions from that initial hypothesis. These predictions take the form of if-then statements. So you would say things like, if all animals need water to drink, then a giraffe needs water to drink. If all animals need water to drink, then a beetle should also need water to drink. And you can test these predictions by just checking to see if giraffes and beetles drink water. If they do, then you have supported your hypothesis through experimentation. If they don't, then you have refuted your hypothesis. And this comes about through a process of data analysis. If you have supported your hypothesis, then you would say the hypothesis is supported and you would continue to do more experiments. If it is inconsistent, you would reject your hypothesis and communicate these results to others. After you've communicated the results, one of the key goals of science is to refine the question and then repeat this entire process. So again, a hypothesis is a testable or falsifiable statement. It is not a question. Hypotheses must be able to make predictions that can be shown to be correct or incorrect through experimentation. Note, however, that hypotheses are never proven to be correct. They are only supported. So in the example that I gave previously, where I said the hypothesis is all animals need water to drink, by looking at several animals and saying all of them need water to drink, we have now determined that at least those animals need it. And it seems consistent across all of the animals that we've studied that they all need it. So we are supporting the idea that animals need water to drink, but we haven't proved that every single animal needs water to drink. Differentiating among plausible hypotheses is one of the main challenges of a professional scientist, and I'll show you what I mean by that here. Imagine that you found a flashlight in your junk drawer that doesn't work. Your observation is the flashlight doesn't work. Your question is, why does this flashlight not work? Well, let's come up with two hypotheses. The first hypothesis is that the batteries in the flashlight are dead, and so there's no energy flowing to the flashlight, and therefore it doesn't work. The second is that it has a burnt out bulb, that the flash bulb in the flashlight doesn't work. How would we test these? Well, hypothesis one, you could test by replacing the batteries. The prediction here is that if you replace the batteries, it will fix the problem. So then we do that, we test this prediction, we replace the batteries, and it doesn't work. So hypothesis one is now falsified. We have said that replacing the batteries does not fix the problem. Okay, so we eliminate then hypothesis one and we move on to hypothesis two, where we test the prediction by replacing the bulb. And the prediction then is that if we replace the bulb, it should fix the problem. And once we've replaced the bulb, the flashlight works. So now we have supported our hypothesis. But a question to stop with here is, have we proved that the problem with the flashlight was the bulb? The answer to this is no. We've supported it, and I think very strongly. This is a fairly simple question, but there are all kinds of things that could have been wrong with the flashlight. For example, there may have been a buildup on the bottom of the bulb that was stopping energy from flowing to it. The bulb might not have been burnt out, it might just not have been working properly. And by changing the bulbs, you fix that problem and not the burnt out bulb problem. Maybe the batteries were seated improperly, or maybe every time you put new batteries in, you put them in the wrong way. There are all kinds of reasons why these things could be different. What we know for sure is that by replacing the bulb, we fixed the flashlight, and so our hypothesis of a burnt out bulb is supported but not proven. The reason why we're being so careful in our language here is because this happens all the time in science. In fact, imagine, for example, that you lived at home last fall and you took a daily dose of echinacea tablets and you exercised every day and you had low stress at school and you had no serious colds. You never had a cold. Versus the fall, where you moved to BU, you stopped taking echinacea, you stopped exercising, you increased your stress at school, and you got sick several times. So the question then is, was it the echinacea that changed your sickness? So was it because you were taking echinacea that you didn't get sick? And in class, I asked the class this question and they said no. But the answer is we don't know, right? We have to test this hypothesis. And the reason why is because you can find all kinds of correlations that do not give meaning. So here we have two different graphs, one for the divorce rate in Maine across years between 2000 and 2009, and the per capita consumption of margarine during the same time. So the per capita consumption of margarine is lowering across this time, and so is the divorce rate. And so we would say that these two things are related, right? No, we would obviously not say that the divorce rate in Maine is correlated with the per capita consumption of margarine, because these two things are likely completely unrelated unless there's something really serious about Maine and margarine. But we could test this. We could test this experimentally. Probably this would be hard to test. It's a social science thing, but you could test your correlations experimentally to test and see if there is a relationship between them. So let's say that we want to know whether or not echinacea does in fact reduce the severity and the duration of cold symptoms. How would we do that? Well, first we need to test the hypothesis. So the hypothesis in this case would be that echinacea reduces the severity and the duration of colds. So we make a control group that we don't treat, an experimental group that we do treat, and we observe the difference between the two. It's important here also that we do this across a large sample size. We use statistical tests to determine if there is a real difference, not just an observed difference. And we also repeatedly test the same hypothesis to determine if we can get the same result multiple times. Okay, so step one, we make an observation. It seems as if people who take echinacea get sick less often, and that if they do get sick, their sickness duration is lower. Then we formulate a hypothesis. Echinacea reduces the duration and severity of the common cold. Then we devise a testable prediction. If the hypothesis is true, then echinacea should reduce the duration and severity of symptoms if taken before a person gets the cold. But actually, we're curious about a couple of different things here. We have to conduct an experiment that tests not only if echinacea reduces the incidence of colds, meaning how often people get colds, but also whether or not it reduces the duration of the cold. So really we need a treatment group of people taking nothing, a placebo tablet, one that doesn't actually have echinacea in it, and a different group of people that are taking echinacea. We have them take this before they get exposed to colds. and then we see whether or not they react to the cold, whether they get a cold or not. And then we don't give them a treatment afterwards. So groups 1 and 2 differ only on whether they take echinacea when they are healthy. If they differ in their rate of getting sick, then that supports the hypothesis. Groups 3 and 4 differ in whether they take echinacea after they've been exposed. If they differ in their rate of getting better, then that supports our hypothesis as well. But you can see how these are totally different treatments. These are different groups. So we need 4 groups to test our hypothesis properly. Okay, so here are the results of our experiment. It seems as if group 2 had the smallest duration of cold symptoms. So group 2 was taking echinacea before. So this seems to support our hypothesis. But the question is, how sure are you that echinacea is the cause here based on these data? And the answer is you shouldn't be very confident here, because the number of participants is really very small. Each group has about 10 participants, which means that in total we surveyed about 40 people for this experiment, which isn't really enough to draw a large conclusion. Think about this picture here of Seth Green. If you were to say, based on this picture alone, how tall are men in relationship to women, you would say men are shorter than women. But on average, men are actually quite a bit taller than women in the United States. The average man is 5'9½", whereas the average woman is 5'4". So this is quite a big difference, and that picture is misleading. So in the same way, if you're looking at our data from the previous slide, if you say based on this very small sample size that echinacea seems to be helpful, then you might be wrong because of the size of the experiment. So we want to be careful with our conclusions and say only that we've supported it, not that we've proven it, as we have before. Isaac Newton, in the same way, did not draw his theory from an apple falling to the ground, right? He drew his theory from a lifetime of experience with gravity that says that when something falls, it will always fall. Biologists also use statistical analyses. We conduct multiple experiments, we combine the data from all of them, and then we compare across these data sets to see how consistent the findings are. Okay, finally, when we go through and we have lots and lots of evidence, a huge body of evidence supporting a particular hypothesis, we would then develop these into theories. Because of the way that science works, as I've said, we can never really prove anything, but we can have overwhelming evidence of the truth of this. So for example, the germ theory of disease. Do germs cause disease? Yes. We can observe that a person with a disease has the germs, and if a person without the disease gets the germs, that they get sick, right? This is a huge body of evidence supporting this theory. And this is what we mean by theory. We don't mean an educated guess here. We mean something that is basically fact, but we can't say is fact because of the way that biology works. The theory of evolution is the same way. We have overwhelming evidence of the theory of evolution. We can do evolutionary biology in the lab. We can create organisms that have different traits than their parents, right? So we know that this is how it works. But it's called a theory because of the nature of science. Okay, so there are 2 key attributes of a theory. The first is that it's consistent with a vast amount of known data. All of the data in the world supports these 2 things. And then it's able to make predictions that are correct regularly. So the germ theory of disease, if a person has the disease, they probably have the germs, and vice versa. This prediction is very robust. Okay, so this is a sort of general introduction to science as it is. Science can be conducted at multiple timescales, at multiple levels of organization, across different organisms, and is unified. Biology is unified by certain principles. And this is how we conduct this science, through hypothesis-driven hypothesis testing, through experimental hypothesis testing. And then eventually we develop all of these hypotheses into unifying theories. that's all that we covered in this first lecture. So I hope this was helpful to you. I'm sorry that the recording didn't work properly. Make sure that when you are clicking on the links, you're clicking on the links for the correct day of the week. In this case, today should have been the Wednesday lecture, and the recording just didn't work properly. So feel free to email me your questions if you were not able to see the recording the first time, and I'll thank you for coming today. So thanks.